Let us first discuss what transposons are. Transposons or transposable elements are segments of DNA found in virtually all cells that move from one location in a chromosome to another within the same chromosome or into another chromosome. It is excised from one site and inserted at another site. By randomly jumping around the genome, they cause mutation and change the amount of DNA in the genome. Transposons were first discovered by Barbara McClinton in maize. The mobility of these genetic elements cause different colors of maize kernels, as you can see in the image. McClinton earns the Nobel Prize in 1983 for her discovery. We now know that about half of our genome is derived from transposable elements. The term transposon was first used in 1974 by Hedges and Jacob for a DNA segment or genetic element which could move from one molecule to another and carried resistance for antibiotic ampicillin. There are two classes of transposons determined by different mechanism of moving within the genome. Class 1 transposons are DNA transposons, which consisting only of DNA that move directly from place to place. Class 2 transposons are retrotransposons, which first transcribe the DNA into RNA and then use reverse transcriptase to make a DNA copy of the RNA to insert into new location. In bacteria, transposons can jump from chromosomal DNA to bacteriophage chromosome or into plasmid DNA and vice versa. Types of transposons. There are two classes of transposons in bacteria: insertion sequences and transposons. Number 1, insertion sequence elements. The simplest transposons are called insertion sequences. The insertion sequence elements are normal constituents of bacterial chromosomes and plasmids. Their name is due to their discovery as insertions within bacterial operons that prevented transcription or translation of the concerned operon or genes. The insertion sequence elements are autonomous units, each of which code only for their proteins needed to sponsor its own transposition. That is, they encode only genes for mobilization and insertion. Physical properties of insertion sequence elements. They range in size from 768 base pair to 5 kilobases. IS1 first identified in E. coli's galactose operon is 768 base pair long and is present with 4 to 19 copies in the E. coli chromosome. The majority of insertion sequences exhibit the short terminal inverted repeats sized between 10 and 40 base pair. These sequences also tend to have RNA termination signals as well as nonsense codons in all 3 reading frames and are therefore polar. Typically, they encode one large open reading frame of 300 to 400 amino acids, and by definition, the protein encoded by this reading frame is involved in the transposition event. Each insertion sequence element is different in sequence, but there are some common features in organization. Now, transposition of insertion sequence elements. Transposition is often referred to as hopping, and this term has the strong connotation that the element leaves one site and goes to a new site, restoring the original site. When an insertion sequence element transposes, a sequence of host DNA at the site of insertion is duplicated. Original copy remains in place. New copy inserts randomly. Insertion sequence element uses host replication enzymes for replication. Transposition requires transposase. The sequence encoding the transposase gene is often located partially within these inverted repeats accompanied by the upstream promoter with the conventional inverted repeat sequences. This arrangement provides the binding mechanism for auto-regulation of synthesis. Transposition initiates when transposase recognizes inverted terminal repeats. The site of integration is called target site. First, staggered cuts are made in DNA at target site. Then, insertion sequence element inserts, and finally DNA polymerase and ligase fills the gaps. Small direct repeats about base pair flanking the target site are created. DNA rearrangement and deletogenic properties of these insertion sequence elements. Insertion sequences mediate a variety of DNA rearrangements. One of the first recognitions of this fact was the involvement of insertion sequences in the integration of F and R plasmids in the host chromosome. This event gives rise to high frequency recombination strains. The initial DNA rearrangement mediated by insertion sequence elements is the insertional duplication that they tend to generate at the site of insertion. IS1 generates 8 or 9 base pair duplication, while IS2 generates 5 base pair duplication. Different insertion sequences are known to cause deletion in neighboring genes and occasionally delete themselves at reasonable frequencies. For example, IS1 causes deletion in neighboring genes at a frequency of 10 raised power minus 3 to 10 raised power minus 4 per generation, and the insertion itself remains intact. These deletions tend to start at the IS1 element, with the other end of the deletion being reasonably non-random and possibly involving some sort of short sequence of homology. It has recently been shown that the position of the insertion sequence related to promoters in the region affects deletion formation by unknown mechanisms. IS2 is similar, but its ability to generate deletions is rather less. Finally, IS4 makes deletions of both itself and DNA on both sides with fair frequency. Number 2, transposons. In general, there are 2 other general classes of transposons. Class 1, also known as composite transposon, are basically the pair of insertion sequence elements flanking a segment of DNA, usually containing one or more genes, often coding for antibiotic resistance. In this class of transposons, the insertion sequence supplies the transposition function and inverted terminal repeat recognize signals. These are essentially insertion sequences wherein 2 copies of the insertion sequence have flanked another gene. Examples are TN5, 9, 10, 903 and 1681. In all of these, the ends of the transposon are capable of transposing separately, since they are in fact subtle insertion sequences. Transposition of IS10 or TN10 occurs at around 10 raised power minus 7 per element per generation. Transposition occurs to a large number of sites in E. coli, but excellent target sites are found at about 1 per 1000 base pairs. Other secondary sites are recognized at poor efficiency. The preferred sites for IS10 and TN10 tend to have the sequence G, C, T, N, A, G, C. Admittedly not very adenine thymine rich, which is then duplicated within the 9 base pair duplication upon insertion. The second class of transposons are known as non-composite transposons. They carry genes, for example, the gene for antibiotic resistance, but do not terminate with insertion sequence elements. Therefore, the ends are non-insertion sequence element repeat sequences. Examples are TN1, 3, 4, 7, 501 and 551. TN3 is 5 kilobase with 38 base pair inverted terminal repeats and includes 3 genes: beta-lactamase, transposase, and resolvase, which functions in recombination. Transposons of this class tend to show regional specificity, and TN3 in particular seems to especially prefer adenine thymine rich regions that have some homology to its ends. Unlike class 1, the ends cannot transpose separately. One member of this class, TN7, has such extreme specificity, that is, it has a single site of preference in the entire E. coli chromosome. It has now been shown that the region conferring this specificity is not the site of insertion but is apparently nearby. A similar separation of recognition and action sites has been seen with certain restriction enzymes. The frequency of transposition tends to be variable, ranging from 10 raised power minus 4 to 10 raised power minus 6. And in the case of TN3, involves a 5 base pair duplication upon insertion. Models of transposition. First model, that is conservative transposition or cut and paste transposition. Conservative transposition is so named because the copy number of the transposon is conserved during the operation. It involves the transposon physically leaving one replicon and moving to another. The original replicon is apparently destroyed, so it is critical that the element transpose to another replicon in the cell. Most elements try to transpose soon after DNA replication, because there is then the highest probability that the cell actually contains another replicon. One thing that is noteworthy that while conservative transposition does not increase the element's copy number in the cell, it does increase the ratio of transposons to replicons in the cell. This might actually be the reason that transposons do not repair the site they have left. The molecular events are illustrated in the image. Double strand breaks occur on both sides of the transposon, coupled with staggered single strand breaks in the target replicon. Then the ends of the transposon are ligated to the single strand segments in the target DNA. Finally, the gaps in the target molecule are filled in. Number 2 model, that is bidirectional replicative transposition. This model differs from the previous one in that the transposon increases its copy number in the cell directly, and the donor molecule is not destroyed. It apparently explains the mechanism employed by TN3. It involves number 1, single strand breaks in the target DNA and on either side of the transposon. Number 2, ligation of ends to produce a complicated hybrid molecule. Number 3, bidirectional replication through the element with subsequent ligation. This results in production of a single replicon via the donor replicon is flanked by 2 copies of the transposon and fused to the target replicon. A replication event resolves this to the finished product. Application of transposons. Number 1, mutagenesis. Since it is clear that transposons transpose to new site at certain frequencies, one might expect that you mutagenize a strain by taking a strain carrying the transposon in a site considered uninteresting in terms of the system being studied, and looking for cases where the transposon has moved and generated an interesting mutation. Number 2, selection for a mutant phenotype. Most mutations leading to the loss of gene function do not have a selectable phenotype. This means that they cannot easily be moved from one strain to another using the gene transfer systems. A mutation caused by a transposon therefore has a distinct advantage since it has 2 phenotypes: the loss of a function of the affected gene and the drug resistance of the transposon. The latter is selectable but brings the former along. It is also noteworthy that a transposon can be viewed as just a selectable marker, allowing linked but not transposon mutated genes to be moved. Number 3, cleanliness. As a mode of mutagenesis, transposons are relatively clean. That is, in contrast to other forms of mutagenesis, they do not cause a large number of alterations in the genome. Number 4, organism specificity. Because most mobile genetic elements borrow some host machinery during transposition, they tend to be limited to hosts with compatible machinery. Most mobile genetic elements found in gram-negative bacteria therefore tend to be specific for that group and fail to function in gram-positives. Number 5, genetically altered transposons. An increasing number of genetically altered transposons are being produced with useful and amusing properties. Number 1, elements with drug resistance of one transposon but with the transposase and the ends of another. Number 2, elements that transpose more or less frequently than the originally isolated version due to differential expression of the transposase. Number 3, elements that transpose only once and then are stable. Number 6, physically detectable homology. When a transposon is used to generate a mutant, the mutated region can be physically isolated using probes for the transposon sequence. The mutated region can then be used in turn as a probe for the wild type sequence. Number 7, evolution of genomes. Since transposable elements cause genetic changes, they make important contributions to the evolution of genomes. The last application, that is not the least, tools for gene cloning and manipulation. Transposons can be used as a genetic tool for analysis of gene and protein function. The use of transposons is well developed in Drosophila, where P elements are most commonly used, and in Arabidopsis and bacteria such as E. coli. I end my today's talk with this note that transposons are the mobile genetic elements, and there are 3 types of transposons in bacteria, which are insertion sequence elements, composite and non-composite transposons. In bacterial transposons, insertion are demarcated by inverted terminal repeats. When they insert into a DNA molecule, they create a duplication at the insertion site. Thank you.